I can see people are starting to join, which is good. Karen, this is a screen live, is it? Yep.
That's correct.
Welcome. As people join and sign on, we'll start in a few minutes, as is tradition. Give it a couple more minutes, I think.
There a bit higher.
Yeah, okay. I think we'll get started and go through the introductions as people are still joining. I'd like to start this session by acknowledging the traditional owners of the land and their elders past, present and emerging. And we pay respects to the activities we perform on this location. It's my pleasure today to welcome you all to this really exciting introduction to technology. We were lucky enough to get some funds to purchase a 10x Genomics Chromium unit, and our staff in the MHTP Medical Genomics are running through the training and operations of that at the moment. I'm, I suppose, like a lot of people, really excited about this technology, which really is appearing in lots of publications and showing us how individual cells interact in complex tissues, both physiologically and the interactions of immune and tumor cells in solid tumors and in sites of infection. So I think it's got enormous potential, and I'm sure will have a rapid uptake. So we're very fortunate that Millennium are going to have agreed to this session today. Both to give us an outline of the single cell genomics and spatial transcriptomics capabilities of their unit, and to discuss the fellowship program they're offering to the Hudson, which will enable sort of people to be trained in this expertise and have ongoing interactions, and also some serious discounts, which is very nice. So it's my pleasure to welcome and hand over to Alex Szabo. Alex is CEO of Millennium Science, and will MC the proceedings today. Thanks, Alex.
Thank you very much, Paul. And good morning to everybody, and welcome. So many of you may know me as originally as the CEO of Jomar Life Research, where in our early days we were probably most well known for providing the free espresso bars at the Lauren conferences, among other events. And now, as Paul's mentioned, I'm also the CEO of Millennium Science, since our two companies were merged last year. I want you to know that it's a really great privilege to speak to you today and to participate in this event, because the Hudson Institute is arguably the single research center with which our company has had the longest and strongest affiliation. Since joining Jomar in 2013, it's really been a privilege for me to support many of you with equipment and reagents to enable the exciting and important work that you do in advancing understanding of basic biology and advancing diagnosis and treatment of disease, and to watch the Hudson Institute continue to develop its international reputation. At the conclusion of the seminar today, I'm going to tell you a little bit about the Hudson Institute Fellowship Program. But as Paul described, this is an exciting program based partly upon our long-standing relationship between Jomar, Millennium, and the Hudson Institute. And to let you know that the Hudson Institute has been selected as one of only 2 research institutes in Australia invited to participate in this unique program, which will offer a broad-based training in 10x Genomics single-cell technology, and offer significant discounts, as Paul has referred to, on the reagents for researchers associated with the Hudson Institute. We're very fortunate to have here today Dr. Jeff Mcdermott of 10x Genomics, who is an Australian, and before returning to Australia in his current role as a science and technology advisor, was actually a co-founder of the company, this world-leading company, which is the leader in the field of single-cell genomics. And he was a research group leader at 10x and was involved in the development of virtually every product that the company currently has to offer. So Jeff has very deep knowledge about how the technology works, and I expect he'll be able to give you, in his talk, some unique insights about how you might consider applying it in your own work. But before I hand you over to Jeff, I just want to express a personal note. As someone who's been involved with many groundbreaking technology companies throughout my career, to say that 10x Genomics is, in my personal opinion, absolutely the most exciting and revolutionary. One of my favorite quotes is from the science fiction writer Arthur C. Clarke, who once said, "Any sufficiently advanced technology is indistinguishable from magic." And I like to think that when considering the capability of 10x technology to profile RNA expression, for example, in hundreds of thousands of cells in a single experiment, I think that magic is a pretty apt description. So I hope that you enjoy Jeff's talk and that it inspires you to imagine new experiments that will take your research to the next level. So please join me in welcoming Jeff. Over to you, Jeff.
Thanks so much for that introduction, Alex. And I'm definitely going to add a magician to my CV now. I like that. So hi all, thank you for coming along today. I just want to start with an agenda for what to expect over the next 40 minutes or so. We'll begin with a general product portfolio introduction and then take a deeper dive into our Chromium single cell and Visium transcriptomic solutions. We'll step through what each of these assay types has to offer and also provide glimpses into our product roadmap that sort of extends into 2022. Although there'll be some data peppered throughout this presentation, the main goal for today is really to introduce you all to the assay capabilities that we offer at Genomics, which of course you all now have access to here at the Hudson. So begin with the product portfolio introduction. And our assays can be broadly associated with one of three major technology platforms. We'll briefly step through each of these, beginning with the Chromium single cell platform. As you can see, this consists of an instrument called the Chromium controller, which is about the size of a shoebox and fits on your lab bench. Into this instrument, you feed these disposable microfluidic chips, and then onto those chips, you can run one of four main assay types. And I'll dive deeper into these assays in the next few slides, but irrespective of the target analyte, the key output from this assay is the ability to characterize cellular heterogeneity. I do also want to highlight the sample format that's required for this platform, which is a suspension of single cells or single nuclei. So that means unless you're already working with cells in suspension, say things like PBMCs, your tissues or organoids will need to undergo some form of dissociation to generate a clean, viable suspension of single cells. The second major technology platform is Visium Spatial Transcriptomics. Unlike the Chromium single cell platform, there's no instrument involved here. So now you're just dealing with this consumable, which is a glass slide that contains a barcoded array on its surface. That's sort of shown by this little cartoon here. Also, unlike the Chromium platform, this technology now uses whole tissue slices as the input rather than a suspension of single cells. So here the tissue is placed on the slide and transcripts are diffused within the tissue down to the surface of the slide where they can be subsequently barcoded. And then following sequencing, we identify what the transcript is. And then this barcode sequence that's appended to the transcript is used to place it back into a 2D grid, giving spatial context to the transcript and ultimately a spatially resolved molecular profile of your image, as sort of shown here. We'll cover Visium in more detail in the second half of the presentation, but let's move to our third major technology platform, which is our in situ product and a piece of technology that I personally find super mind-blowing. Just like the Visium platform, here the input into this workflow will be tissue sections. However, on this platform, you're now detecting mRNA molecules directly in the tissue itself with either cellular or subcellular resolution. So rather than extracting mRNA molecules out of the tissue, barcoding those molecules on a 2D array, sequencing those barcoded transcripts, and then using that barcode to place the transcript back into its spatial context, here we're just detecting the presence of the mRNA molecule within the tissue itself. That gives rise to data such as this. Here's an outline of a mouse brain sample showing the entire image tissue. The color is a powerful representation of how rich the data is. And this becomes even more apparent when zooming in and focusing on specific cellular markers. Here at high magnification, you can appreciate the single molecule resolution of the in situ assay, with each symbol representing an individual RNA molecule. However, since this is a product that's still under development, I won't talk about it sort of beyond this example today. But if you do want to know more or kind of keep up to date with our advancements here, please just reach out and happy to share as stuff comes to light. But for today, I want to refocus our attention back to the Chromium single cell and Visium spatial transcriptomics platforms and briefly step through each of these assay types. So as I said earlier, this technology platform consists of an instrument called the Chromium controller, which is about the size of a shoebox and fits on your lab bench. And into this instrument, you feed these disposable microfluidic chips, and then onto those chips, you can run one of four main assay types. Although all these assays have different output, since they all use the same instrument and fundamental microfluidic architecture, they all broadly have a similar workflow as depicted here. Rather than step through the workflow in detail, in the interest of time, I really just want to highlight that for all the assays, we have ready-to-use robust workflows, including demonstrated protocols, various sample types, and freely available turnkey software tools. So if you're new to the world of single cell and want to apply it to your research, but feel sort of maybe somewhat intimidated about where to begin, then we have solutions across the entire spectrum of the workflow to really enable you to be successful in your single cell experiments. The other thing I wanted to quickly highlight before we step through the output from each of these four assays is the launch of our new Chromium X instrument. Just like its predecessor, the Chromium X is a benchtop instrument as well, that you also run all of our four main assay types on. However, the Chromium X now comes with the ability to run high throughput versions of the single cell gene expression and single cell immune profiling assays. And we've increased the throughput of these two assays in two key areas. So the first is by doubling the number of samples you can run per chip. So on our standard throughput, you can run 8 samples or up to 8 samples per chip. On our high throughput, you can run up to 16 samples per chip. The second is doubling the number of cells you can capture per sample. So on our standard throughput, you can capture up to 10,000 cells per sample. And on a high throughput, you can go up to 20,000 cells per sample. And then there's ways to increase sort of beyond those numbers as well using sample multiplexing. So in total, there's a 4x increase in cell throughput if using high throughput assays on the Chromium X instrument. And apart from the obvious cost savings associated with high throughput assays, I really want to mention them as they pair very nicely with many sort of functional genomic studies. You know, for example, if you're performing organoid drug screens, you have the capacity to profile all the samples simultaneously within a single run of the instrument, or if you're performing things like pooled CRISPR screens, you can now screen many more guide RNAs per pool. Let's refocus our attention back onto those 4 main single cell assay types, and importantly, what the output from each is. And we'll begin with our single cell gene expression kit. The target analyte from this solution is mRNA, where we're capturing transcriptome-wide 3' digital gene expression from single cells. So that's definitely the core of the assay, but we can actually extend the menu of analytes being detected from a single cell using what we call feature barcoding technology. So you can also detect cell surface proteins in addition to transcriptome-wide gene expression from the same single cell. This is achieved by having a DNA barcoded antibody, which binds to a cell surface protein. And really, this is akin to FACS, where in FACS, you use a fluorophore barcoded antibody to detect cell surface proteins. However, now in our single cell realm, we're using a DNA barcode rather than a fluorophore. The number of cell surface proteins that can be multiplexed is essentially unlimited, as you're no longer limited by the spectral overlap of dyes in the same way that you are when you use FACS. There is, of course, definitely some practical limitations to just how many cell surface proteins you can measure, but we do see commercially available panels from our third-party vendor BioLegend of well over 100 antibodies specific for different cell surface proteins, sort of being routinely used out in the literature. You know, we are supplied as pre-titrated lyophilized panels that you can essentially buy off the shelf and use straight away. Using feature barcoding, you can also detect CRISPR perturbations alongside transcriptome-wide gene expression from the exact same single cell. And in fact, you could even detect cell surface proteins on top of that if you really wanted to. This works incorporating by a small piece of DNA into the guide RNA itself, so it can be captured by the assay biochemistry. So you get a readout of what the guide RNA was, which tells you about the CRISPR perturbation that occurred in that single cell, but you're also getting a transcriptome-wide snapshot of the functional impact of that perturbation, again, linking the guide RNA to the transcriptome-wide gene expression from the exact same single cell. There are many applications of single cell gene expression. I guess I've chosen to highlight this one, really because I find it quite a powerful study. It was used to provide actionable insights into lung tumor microenvironment. So here, 5 patient tumor samples and their matching non-tumor samples were taken directly from the operating room and turned into single cell suspensions and ran through the Chromium instrument. So why did they do this? They wanted a comprehensive catalog of the tumor microenvironment transcriptome in human lung tumors. So by doing this, they identified 52 stromal cell subtypes, including novel subpopulations that allowed them to elucidate differences between normal and tumor-derived T cells, and ultimately identify new targets for immunotherapy. So again, I really like this study, and it's just a real brief skim through of it there. That's sort of the high-level details, but taking a patient sample directly from the operating room onto the instrument, not too long later, a couple of days later, gaining actionable insights and identifying new targets for immunotherapy, I think is a pretty powerful story. So, as I said, single cell gene expression has been used across a variety of different applications. However, one interesting niche that I definitely did want to mention as well is that single cell gene expression kit is becoming routinely used for working with organoid models. The single cell measurements in this context make so much sense as they provide deeper insights into lineage tracing and organ developing systems well suited for high throughput drug screens, modeling human disease, or even just for validation, such as determining what cell types and states a culture is comprised of. We'll look at one of these applications in more depth, but this body of work is definitely growing every day. This preprint is a fantastic example of how organoids can be used for drug screening, and in this case, personalized drug screening, and that comes from the University of Utah. In order for us to understand cancer biology and develop more effective therapies, we need a model system that can recapitulate the complexity of human tumors and the variable treatment responses to different drugs. This publication demonstrates the feasibility of development, characterization, and utilization of patient-derived organoids from large banks of primary cancer samples. And we're looking at breast cancer in this publication. When you have paired patient-derived xenografts and patient-derived organoid models, you can also perform matched in vivo and in vitro experiments, and even perform treatment dose response testing on the organoids initially in vitro, and then confirm the findings in vivo. And the authors here use single-cell gene expression product to profile transcriptome-wide mRNA from single cells, and their results show that the paired patient-derived xenografts and patient-derived organoid models largely recreate the mutational environment of the original patient tumors. Let's take a look at our single-cell immune profiling assay. Just like the previous example, the core of this assay is detection of mRNA, but now we're capturing whole transcriptome 5' digital gene expression from single cells. And because we're capturing the 5' end of transcripts now rather than the 3' end, we can expand the menu of analytes to now include full-length paired VDJ sequences from both T and B cells. The way this works is that you simply perform a PCR enrichment step from the whole transcriptome mRNA molecules that were captured from single cells halfway through the workflow, and you simply pull out the VDJ sequences. This means that there's no computational wizardry here to do the pairing of alpha and beta chains if you're looking at T cells, or heavy and light chains if you're looking at B cells. You're detecting exactly what the cell is making, so the pairing information comes from the cell itself. You can also detect cell surface proteins in addition to transcriptome-wide gene expression and cell surface receptors, again, all from the same exact cell. Whilst the oligo barcodes that are added to the antibodies for this assay are different in structure to the 3' assay, meaning that the antibodies are not interchangeable between the assays, the barcoding premise is still the same as what I described previously. And now what's super cool is on top of all this information you can capture from the exact same single cell, you can also layer on antigen recognition. Just like the other feature barcoding assays, this works by applying an oligo barcode indirectly to your antigen of interest. So when that antigen binds a T cell or a B cell surface receptor, it remains bound, and this oligo that is present can now be picked up by the assay biochemistry and used to infer the molecular identity of the antigen species present. Another quick literature example on this topic in a few slides. So from just one assay, you can get whole transcriptome digital gene expression, full-length paired VDJ sequences, cell surface protein, and antigen recognition, all from the exact same single cell. To give you an appreciation of the power of what is possible using the single-cell immune profiling kit, I just want to step through some single-cell data that we generated in-house at 10x Genomics. In this experiment, we took some PBMCs and labeled them with antibodies specific to 17 different cell surface proteins. This immunology panel of antibodies was purchased from our third-party partner, BioLegend, where all the antibodies have been pre-titrated and supplied as a lyophilized mixed. I should say that this is a pretty modest-sized antibody panel, but you can see in comparison to, say, a 6-color fax experiment, just how much extra information we can already get by just using the sequencer as a readout rather than fluorescence. So we stained this out, so the cells were stained with this antibody panel, washed, and then ran through the Chromium controller. And we generated data for whole transcriptome digital gene expression, full-length paired VDJ sequences, and cell surface protein. The first piece of data to consider is whole transcriptome gene expression. Here, the t-SNE projection that's shown, the single cell data, where each dot here represents a single cell from the experiment. And using canonical cell markers, we can broadly identify the major subclasses of cells present in our PBMC population, from T cells to B cells and macrophages. And you can see that legend over there on the right. But remember, we also collected cell surface protein information alongside whole transcriptome gene expression for every single cell. So what we did was recluster our cells based entirely on the cell surface protein information, and then used our annotations from the gene expression data to more deeply characterize the PBMC sample. And you can see the t-SNE projection on the right here as our populations are more defined, as we have deeper insights by measuring both protein and RNA. This can clearly be seen in the T cell population, where we can now begin to discriminate or separate memory from naive T cells. It also goes to show the power of a multi-omic approach, where species present at the protein level might not be reflected at the transcriptome level, and vice versa, giving reason to why you'd want to measure both protein and RNA from the same single cell. Moving on now, we also collected full-length paired VDJ sequences from our T and B cells. We'll begin with T cells, where in the middle here we have our t-SNE plot, where cells have been clustered based on their gene expression profile, so the same as what I just showed you. But now each blue dot represents a cell where a full-length paired T cell surface receptor was identified. Over on the right panel, you see we have a list of the top clonotypes present in the sample, and then on the left we have an actual mRNA sequence for both the alpha and the beta chain for one of these clonotypes, just as an example. But of course, we get that for every single clonotype here. And then we, of course, extract the same information from our B cells. Again, this is the same t-SNE projection, but now each blue dot here represents a cell where a full-length paired B cell surface receptor was identified. And then again, over on the right panel, you see we have a list of the top clonotypes present in the sample. And then on the left, we have the actual mRNA sequence for both the heavy and the light chains for just one of these clonotypes, as an example, but again, you get it for all the clone types that were detected. So as sort of a topical example, we're seeing that single-cell immune profiling kit being extensively used in COVID research, both for better understanding the disease, along with measuring vaccine response to the disease. However, since like all of you, I'm pretty tired of hearing about COVID, I won't choose any of these specific papers to dive into, but I did want to pause and highlight the significant advances in our understanding of both the disease, along with how people respond to the corresponding vaccine that have actually been made using the single-cell immune profiling kit. I've been definitely quite proud to see our tools being used in such groundbreaking ways. So rather than a COVID paper, I chose this really nice piece of work to further highlight the power of our single-cell immune profiling kit. It is entitled High Throughput Mapping of B-cell receptor sequences to Antigen Specificity, but some of you might actually know this as LibreSeq. The authors used single-cell immune profiling kit for the simultaneous measurement of whole transcriptome gene expression, full-length paired B-cell surface receptors, and importantly, antigen mapping using that feature barcoding approach I talked about a couple of slides earlier. The reason they chose this multi-omic approach was to map paired, heavy and light chain B-cell surface receptor sequences to their cognate antigen specificities from thousands of B cells derived from 2 HIV-infected subjects. So they're really mining these subjects to find novel, broadly neutralizing antibodies. Here's a high-level snapshot of the LibreSeq workflow. Basically, PBMCs are mixed with the DNA barcoded antigens. You then sort for B cells that are bound to antigens, so you enrich just that population. Then you simply run this enriched population of antigen-bound B cells on the single-cell immune profiling solution. It is a note, this is a customer-derived workflow, but at 10x we'll have a commercial version of this assay next year, which we call Beam. This means if you want to run this in your lab, we'll have a solution for you that should cover everything from sample prep to analysis, and really you should just be supplying the antigens, and we'll have the rest of the workflow for you to complete these experiments. And then this is a cross-reactivity plot. I think it's really worth diving into a bit because these plots are basically unprecedented prior to LibreSeq. So keep in mind that B-cell receptors can bind more than one antigen because of the variable region maturation. The epitope that is recognized by the B-cell surface receptor may have common binding to multiple antigens. So the 5 barcoded antigens mapped in this patient's serum are shown below on the bar chart. If the isolated B-cell contained a sequencing read for a given antigen, the circle is colored black. And then from left to right are B cells that bind increasing numbers of antigens. And the bar chart itself is the number of times this B cell configuration is counted. This graph is critically important because it shows how broad the antibody binding is. This is not based off convoluted bioinformatic algorithms from sequencing bulk B cell DNA. These are physical counts of real associated bound antigens. And again, this was sort of unprecedented prior to this study. So from this analysis, they actually identified a previously unknown broadly neutralizing HIV antibody from a HIV positive donor. So let's switch gears a little bit now and talk about our single cell Ataxic assay. Instead of mRNA, we're now detecting DNA in the form of genome-wide open chromatin regions. This is really our first foray into the world of epigenetics. And the open chromatin information reveals areas of active gene transcription, regulatory regions, and binding site motifs. Just got some example data that, again, we generated it in-house at 10x Genomics for approximately 10,000 PBMCs. You can see using that chromatin signature, we can classify our subpopulations of cell types that are present within our PBMC sample. We can kind of go a little bit beyond traditionally what we do with gene expression. So gene expression we're counting is a gene present or absent, but here using our Ataxic profile, we can look at the gene regulatory environment to sort of help us better define our different subclasses of cells. And I think this is a nice example of that. So what we've got here is a plot for the CD4 gene and our different major subpopulations of cell types, their Ataxic track, if you will, separated out. And you can see the different peaks around the gene. So basically, again, in our single cell gene expression information assay, we capture information whether is CD4 present or absent. But here we're looking, what are the gene regulatory environment around the CD4 gene? So we can do things like look at the promoter accessibility in T cells, and you can find it in T cells and monocytes. So a strong peak in open chromatin region there in the promoter. You can also look at the repressor region. You can see strong open chromatin peaks in the repressor region for monocytes and dendritic cells. We can look then also in the enhancer regions where we find it only in CD4 T cells. So moving on to our last assay, which is our Multi-Msa assay. The moment we launched our Ataxic assay, everyone understood the power of the measurement, but also immediately jumped on what if I could get both epigenetic and whole transcriptome information from the exact same cell. So they really wanted to look at DNA and RNA, but they wanted it from the exact same cell. So that's where this product was born from, our single cell multiome kit, which is really an amalgamation of our Ataxic and 3' gene expression assays. And again, it allows you to identify both open chromatin and transcriptome-wide gene expression and link that to the exact same cell, which really allows identification of putative regulatory elements that you can directly link to a gene of interest. So this is some more data generated from in-house at 10x Genomics, again, looking at PBMCs, about 10,000 of them here. And we begin by looking at the gene expression. So again, you can see here's our gene expression clustering. We're looking at RNA level now. We can broadly classify major cell subtypes that are present. We can do the same thing for our Ataxic signal. This is this same population of cells, just looking at open chromatin regions. And again, we can broadly classify our major cell populations. But what we have now is the power to link these two modalities together from the exact same single cell using our barcoding information that we applied to it. So we call it inference-free linkage, the transcriptome and epigenome across thousands of cells. There's a quick literature example, I think nicely highlights the power of simultaneous attack and mRNA measurements from the same cell. I've selected the work from researchers at Mount Sinai here. Here they used a multi-omic kit to deeply study a population of human pituitary stem cells, which are important in tissue homeostasis and renewal, but are incompletely characterized. What you can see in the UMAP plot here on the top right is the clustering of different cell types identified in the human pituitary gland based on their combined gene expression and attack signals. So the top UMAP plot there is your multi-omic plot, if you will, combining both those modalities to be able to nicely separate those major cell populations in your sample. But since the researchers were primarily interested in the stem cell population, then they reclustered just these cell types. And that led to the UMAP plots along the bottom, where at the mRNA level, they observed differential gene expression patterns in this apparently homogeneous population of stem cells. So just because the cells are the same type doesn't mean they express exactly the same genes. But the key question is what mechanisms are leading to differential expression within this seemingly same population of stem cells. So let's quickly examine how they use the multiome data to provide insights into transcription factors and epigenetic mechanisms that shape gene expression within the same pituitary stem cell types. First, we can examine the gene GATA3, where the authors actually found no change in transcription factor expression between the stem cells, so no changes at the RNA level. However, they did observe significant change in the chromatin accessibility across the stem cells, as highlighted by the red bars on this plot on the left. This suggests that the chromatin accessibility is the predominant driver of the GATA3 differential expression within stem cells. The next example is the Pomc gene. Unlike the previous example, here there was actually no change in chromatin accessibility. So that's the plot on the left down the bottom. However, at the mRNA level, there were significant differences in expression of two key transcription factors, as shown in the blue box. This suggests there's differences in transcription factor expression as the predominant mechanism of differential expression of this gene within stem cells. They can identify it's the RNA level, not at the DNA level. And hopefully this little example really highlights the power of single cell sequencing as necessary for studying heterogeneous, or even in this case, supposed homogeneous populations of cells. And what is more, multi-omic single cell approaches such as those highlighted here provide rich insights into any biological system. So I want to round out this section with mentioning some new products for the Chromium single cell platform that we have coming. Available now, which is products we launched this year, we have targeting panels, Cellplex, and low throughput kit, and actually just our Chromium X high throughput. So targeted panels are exactly what they sound. It allows you to still capture the whole transcriptome gene expression, so you get to the very end of the workflow. But rather than sequencing the whole transcriptome, you can actually do a weighted pull down of just the genes of interest that you care about and only sequence those. So it's sort of a cost-saving initiative, if you like. If you're doing larger studies and you don't necessarily need the transcriptome-wide snapshot of the single cells, you've got panels of thousands or even hundreds of genes that you really care about, and that's going to be informative for you. You can pull those genes out of your whole transcriptome libraries. You've still got that information, but when it comes to sequencing, you're only sequencing a fraction of it and you're saving some money. We have our Cellplex kits. These are another cost-saving initiative from 10x Genomics, and it allows sample multiplexing. I mentioned it earlier on in the presentation, but essentially what you do here is you apply a DNA label to each sample during the sample preparation process. And this allows you to pull those samples together and run them as one meta sample, if you will, through a lane of a Chromium control on one of the chips. You can then bioinformatically tease apart which sample is which based on this DNA barcode that you applied at the beginning. And again, this is a cost-saving initiative in that allows you to combine multiple samples into one channel, so you save some money on the 10x reagents. You still have to do all the sequencing, so we don't save anything there, but you can combine these Cellplex experiments with targeting panels to save even more, so you can save money on the 10x, then do targeting and save money on your sequencing. Low throughput kits, exactly what they sound, enable measurement of 100 to 1,000 cells. It's a cheaper kit, and the point of this kit was really twofold. One, if you're looking at pilot studies on a really limited budget and you want to apply for grants, you can kind of purchase this and get a small amount of single cell data to justify why you get money for more. Or importantly, if you're doing some upfront sample prep optimization that really requires the single cell sequencing readout as the way to inform your optimization processes, then you can run a low throughput kit, do some small amount of cells, small amount of sequencing, all for lower costs before running a much larger study. Chromium X, as I talked about before, this is now, we can run all the assays that I just talked about on that instrument, but also high throughput versions of the single cell gene expression and single cell immune profiling, really for those people who are looking to take it to the next level and perform much larger experiments. And this is just an economical way of scaling. And then we have our 3 products coming next year in the Chromium single cell space. So we have our fixed RNA profiling kit, and I've got a couple of slides on that next. 5' CRISPR, so we're going to enable CRISPR measurements in the single cell immune profiling assay as well. So this is pretty cool because it's not going to involve redesigning your guide RNA molecules as you have to do in the 3' assay, because you need to put a small piece of DNA in those guide RNA molecules in order to capture them. Here, you can leave the guide RNA alone, and we provide a primer that you can add in order to capture that guide RNA. So just another modality you can add on to, the 4 or 5 modalities you can already get out of this assay. So it's pretty cool to see. And then we have our barcode-enabled antigen mapping, or BEAM application that I just talked about previously with the LibreSeq. That's going to be our commercial version of that assay, and it'll be applicable to both antigen specificities or antigen binding for both B cells or T cells as well. But I do want to quickly mention the fixed RNA profiling kit, just because that is coming very soon in the next couple of months. So a little bit more about what it is, and really as single cell researchers using clinical samples has grown, there's a new challenges with patients spread across the country or globe, samples being collected at all hours, and fragile cell types that can decompose before they make it onto the Chromium instrument. A new single cell fixed RNA profiling kit will help overcome these challenges. Paraformaldehyde fixation will make it possible to stabilize samples at the point of collection, preserving biological states. The workflow will be compatible with any single cell suspension. In addition, the sample can be optionally stained with antibodies for compatibility with cell surface protein measurements prior to fixation with PFA. And the assay is designed to capture either a subset of genes via targeted panels or whole transcriptome, and achieves gene detection sensitivities comparable with fresh samples. And you can see that on the next slide here. So I've got a tiny snippet of data from R&D where we're comparing results from PBMCs run either fresh on a single cell 3' solution or after fixation using the fixed RNA kit. When we started developing this workflow, our goal was to achieve comparable sensitivity to our single cell 3' V3 gene expression solution on fresh samples. So here we compare UMI detection over our 2000 gene immuno-oncology panel, where you can see that sensitivity is similar between the fresh 3' control and the sample progressed with the fixed RNA profiling kit. So that's the plot on the left, and you basically see those two lines are very closely matched on top of each other. Cell type annotation is also similar between the two samples, which is great to see. We're preserving the biology, but also fixing the sample. So to reiterate, this product is coming very early next year. So if you're thinking these samples or study design may not be amenable to single cell sequencing, then definitely come talk to us. This product will likely overcome many of the challenges that you might be facing. So I want to change gears now to our Visium spatial gene expression product. I'll begin with the first sort of high-level question, which is why spatial analysis? And it's really because location matters. So in this example here, we have a hot tumor and a cold tumor. So at the top, our hot tumor, and you see on the left is a H&E image of that, and on the right is an immunofluorescence image where we're looking at tumor infiltrating lymphocytes. And at the bottom is our cold tumor, again, H&E image on the left, and our immunofluorescence image on the right, looking at tumor or looking at lymphocytes. Top is associated with good prognosis. You can see those lymphocytes have infiltrated the tumor and able to do their job. And on the bottom, we have a poor prognosis. You can see those lymphocytes are located in a ring around the outside of the tumor. So we can definitely do single cell sequencing on this sample, but remember we have to take it and dissociate it into the suspension of single cells before running it on the platform. So when we do that, we lose all of this spatial context, and we might be able to tell at the expression level what the difference between lymphocytes and the top and the bottom example is, but ultimately, you know, we have no insight into their spatial location. So that's really where our Visium product was born, to allow the combination of those two measurements. To sort of step through the data that you extract out of the Visium workflow. So we of course begin with our histology image, or H&E. So this is a mouse brain, and you know, you still capture an image of your tissue. So whether it's H&E stained or immunofluorescence, and we support up to 6 colors, which really means sort of 5 targets plus a DAPI control. You can still retain that very important histology image. So you can still do you know, cell type annotations and really, you know, the visual inspection of your tissue. Typically, obviously, that's where your journey ends, though. But with Visium, we take it the next level further, where we capture the underlying whole transcriptome gene expression information from this tissue. So we're really able to extract at the molecular level our information beyond just at the visual level. In the previous examples I showed you, you know, each individual spot, each individual dot in these plots represented a single cell. Whereas here in these examples, each individual dot represents a spot. And I'll explain more in some upcoming slides about what a spot is, but I think it's pretty evident here when you look at this data as to what a spot is. But basically, this is where the power of Visium comes into its own. We can take that underlying whole transcriptome gene expression information and add spatial location back to it and really pair it with our visual image of the tissue to see, you know, what's happening in different regions, both transcriptionally and visually. So just like our Chromium single cell platform, we have ready to use robust workflows for this, including demonstrated protocols for various sample types and turnkey software tools. And we have 2 flavors of this kit. So we have our fresh frozen or our FFPE. As the name suggests, it just depends on which tissue type you're working with as to which kit you would select. They both have a sort of broadly same high-level workflow as depicted here, where you section a tissue onto a slide, you get your imaging of that tissue, and then you complete your molecular barcoding on the surface of the slide and sequence that, and you do the data analysis. But there are some fundamental differences between how we detect transcripts in these kits, and I'll expand on that in a minute. But I do want to dive into, I guess, the technical specifications around the Visium slide itself. So here's a cartoon of the slide. You can see there's 4 capture areas per slide. More importantly, we have to run all 4 capture areas at once. You can see each capture area is 6.5 by 6.5 millimeters in size. Importantly, your tissue has to fit within this capture area. So you see a red border around here. This is a fiducial border we actually use for alignment between the image you take of your tissue and the sequencing data. It's important that there's some of this border preserved. Your tissue can overhang a little bit on either side. You know, it's common, but really the big, I guess, failure that happens is when we automatically process the data through analysis pipelines, you might not be able to automatically align the image. So you might actually have to manually go in there and do that if you do sort of cross over these borders. It's not the end of the world, but ideally you're only generating data from within that 6.5 by 6.5 millimeter array. So you can see within there, as I mentioned, is an array. It's approximately 5,000 barcoded spots, where each spot is approximately, or is 55 microns in diameter with 100 micron center to center distance. So this is important because it's not quite single cell resolution yet. I'll have a note on that in the next slide. So 55 microns, depending on your tissue type and cell sizes, roughly, you know, anywhere from, you know, 1 big cell right up to maybe 5 or 10 cells per spot. So, you know, it's important to keep that in mind when running Visium experiments. As I just showed on the previous slide, those t-SNE projections of the gene expression, each dot represents a spot. So each dot represents, you know, a handful of cells now, not a single cell. And importantly, each one of these barcoded spots here is really a lawn of oligonucleotides. I won't go through the exact structure here of them, it's shown on the right, but the important part is that each one contains a spatial barcode where we know the spatial barcode for each one of these spots. So when we append this spatial barcode to our transcripts, we sequence the transcript to identify what it is, we sequence the barcode that's appended to it, and that allows us to place that transcript back into this 2D grid here. A quick note on resolution. as I just mentioned, our product at the moment has 55 micron spot size, which you'll see in a second is really sufficient to generate some pretty nice spatial transcriptomics data. But certainly, the second we launched this product, we're a single cell sequencing company, and everybody asks, well, where's the single cell version of it? So we're working on it very hard in R&D. Something that's coming mid next year, we believe. We believe everything's on track for that launch. And we call it Visium HD, where the resolution has been really dialed down to just single micron resolution. I believe it's on the order of 4 to 5 microns. So you can see now, really capturing single cells. So quick note on this, as I mentioned, it's coming mid next year. A lot of people say, well, I really will wait for it. I'd sort of challenge that, if you're interested in spatial transcriptomic experiments now, think about Visium in its current format. A, Visium HD is going to be more expensive. B, it's going to require more sequencing. C, it's not quite out yet. And D, you can get up and running with Visium as it is now, kind of overcome this technology adoption curve where you can understand how your sample works, get the data, get your pipelines up and running for the data, so that when Visium HD is launched, it's kind of a drop in into your lab, and you're not going to have to wait a couple of months to figure out how the technology works. You'll already be familiar with everything, and it should just be basically a new slide in your lab that you process no differently to the ones you've been processing now with Visium. So a quick note on the two different kit structures. As I mentioned, there's fresh frozen or FFPE kits. So for the fresh frozen kit, what we do is we place the fresh frozen tissue that's been embedded in OCT directly onto the surface of the slide. We then permeabilize that tissue, and then the mRNA molecules within that tissue diffuse directly down to the surface of the slide where we barcode them. So fresh frozen, we're detecting mRNA or anything polyadenylated, basically, directly from the tissue itself. And because we're detecting anything polyadenylated, it's applicable to a wide variety of species, basically anything that's got polyadenylated transcripts. We even see it being used in plants. However, for the FFPE kit, it's slightly different. FFPE process itself damages mRNA molecules, so we can no longer detect them directly. We found we tried to do that, but the sensitivity of the assay was really poor and was really affected by heavily cross-linked molecules. We just couldn't extract the information out. So in this kit, what we do is you take your fixed FFPE fixed tissue, you still apply it to the surface of the slide, but now you actually flow on probes onto that tissue, where we have a left-hand side probe and a right-hand side probe, and they anneal to the transcript if it's present, or if the transcript is absent, those probes are simply washed away. We ligate the left-hand and right-hand side probes together to lock in their specificity. And then what we do is we actually permeabilize the tissue and diffuse the probes themselves down to the surface of the slide rather than the transcripts. So we barcode probes and we sequence probes in the instance of FFPE. So we use the probe as a proxy for the transcript. Because we're using probes, we have to use probe sets. And we've developed probe sets for human and mouse. But if you're working with FFPE tissue from other species, then unfortunately we don't have those panels ready just yet. And now I'm going to move on to actually looking at an example of some data generated using the Visium platform. This data vignette comes from work that was done directly in-house at 10X, where we studied the tubergenesis of triple positive ductal carcinoma using spatial and single cell assays. Triple positive ductal carcinoma is a precursor lesion that can give rise to invasive breast carcinoma, and elevated expression signatures of HER2 and the estrogen and progesterone receptors are the hallmark of this cancer type. We profiled the transcriptome, the tumor microenvironment, for a 2-year-old frozen biopsy sample using 3 complementary assays. So we did it on Visium FFPE, Visium fresh frozen, and our single cell gene expression. So the way we did this is basically one half the sample was fixed, embedded for FFPE, and stored at room temperature, and this was used to run on the Visium FFPE. The other half of the sample had been snap frozen and stored at -80. So that half then we split into 2. So it would be like a quarter of the sample was run on the Visium fresh frozen, and the remaining quarter was nuclei extracted and then run on the Chromium single cell platform. So the first piece of data examined is the comparison between fresh frozen and FFPE Visium spatial transcriptomics results. And here on the left, we have a H&E image taken of the FFPE sample. Then these 2 plots on the right show how the spatial data from each kit is correlated. And as you can see, there's a high concordance between the 2 data types, with both kits providing similar insights from the same sample. And this is really great to see, especially as one result is generated by detecting transcripts directly from the fresh frozen material, and the other is from detecting probes that are added into the FFPE material. So I've definitely, the plot on the right-hand side where fixed has a little lower sensitivity than fresh frozen, I've definitely seen this switched around the other way where fixed has a little higher sensitivity than fresh frozen. But the key take-home message is that there's no degradation in performance when using the FFPE kit because we've switched to that probe-based system. We asked the pathologist to annotate the cancerous regions of the sample, represented here by the outlines in these images. So now you have a grasp of where the cancer cells are located, but is there more biological information hidden in this tumor? And I'll just quickly highlight that you don't need these annotations to do the spatial transcriptomics workflow. Just in this case, we did it to see how the annotations aligned nicely with our transcriptomics data. So let's investigate further. First, we'll look at the clustering. Using Visium for FFPE in combination with our Space Ranger software, we can output 4 major clusters based on differential gene expression. And you can see how tightly these clusters align with the pathologist annotations. For example, the red cluster identifies regions of carcinoma within the tissue. If we use immunohistochemistry to look at the diagnostic markers of triple positive breast cancer, HER2, and the estrogen and progesterone receptors, you can see the Visium gene expression data correlates very nicely with the protein data. So along the bottom here, we're looking at the protein level, and at the top is our Visium data. So we're looking at those same proteins, but at the transcript level. You can go much further with Visium data, subdividing these larger, apparently uniform areas into regions with distinct molecular signatures and identifying clinically relevant cell types. So that's sort of what's shown on the right-hand side here. You can move beyond just those 4 major subpopulations and really do unbiased clustering to identify, regions with distinct molecular signatures and identify clinically relevant cell types. Let's look at the pathologist annotations for another sample and focus on the regions of ductal carcinoma highlighted in yellow. Now we'll add our Visium gene expression information, and you can see yet again how well this data aligns with the data provided by the pathologist. We'll go a step further, as you don't have to stick with just a few markers, but you can investigate the entire transcriptome. Let's answer the question many of you are thinking at this point, which is where are the immune cells? So tumor infiltrating lymphocytes are the hallmark of the immune response in cancer and have prognostic value in the clinic. And here's an example where we use Visium FFPE to identify infiltrating T cells and macrophages. So if you think back to that initial example I talked about at the very start of the section where you could see different lymphocytes and how they are either infiltrating or not, we're doing the same thing here, but using our spatial transcriptomics data to really visualize where those cells are rather than just picking them. It's also possible to drive powerful analysis of the Visium data by combining it with data from our single cell assays. We paired our spatial data with single nucleus RNA-seq data generated using our Chromium single cell 3' assay. Cell type expression profiles derived from the 3' assay allow us to estimate cell type proportions for each Visium feature and give insight into cell type co-localization. And as an example, staying on the topic of immune infiltrates, we can map B cell populations at different stages of maturation within our breast cancer tissue section. And here we see the distributions of memory B cells, plasmablasts, and then the mature plasma cells. So realistically, what we're doing here is we're building up a profile of those 3 cell types from our Chromium single cell data, and then we're hunting for them in our Visium data, where again, it's not single cell resolution. We can break down, however, the profile of each spot to find the probability of our cell types being there based on the expression profiles that we're deriving from the single cell data. So to round out today's talk and this section, I just want to mention some new products that we have coming in the realm of spatial genomics. So we've made some significant advances in our spatial platform, including Visium FFPE that was launched this year. But we have, and I apologize, the dates on this might actually not be as accurate as they should be, but these are what we have coming sort of into next year. So we have larger capture area slides. So instead of a 6.5 by 6.5 millimeter array, they're actually going to be 1 centimeter by 1 centimeter arrays. And you can see there's only 2 of them per slide now, so 4. We have highly multiplex protein, CytAssist, and Visium HD coming. And with the final minutes, I'll quickly talk about the highly multiplex protein and the CytAssist. And of course, the Visium HD we already covered coming in mid next year. This is going to be single micron resolution Visium slides. Let's begin with our highly multiplex protein detection. Our current Visium product already allows for the co-detection of a handful of proteins using immunofluorescence. But our goal is to enable looking at even more proteins at the same time, simultaneously capturing gene expression from the same tissue slice. This is why we'll introduce our highly multiplex protein solution for Visium. Just like immunohistochemistry, protein detection is still based on using antibodies, but instead of antibodies being labeled fluorophores, now they'll be labeled with DNA barcodes. And just like our other examples of cell surface protein, this significantly increases the multiplexing space to allow you to measure protein expression from tens to thousands of proteins. And of course, you get whole transcriptome gene expression from the exact same slice of tissue. Here's just a quick snapshot of some data. On the top left is the immunofluorescence signal for 2 proteins, CD29 and CD4. And on the right is our Visium product, where we're looking at the detection of 12 proteins from the same section. These proteins are detected using antibodies, but those antibodies contain a DNA barcode, so we can use our next-gen sequencer as the readout. Just remember, I'm showing only protein data here, but of course, we do capture whole transcriptome gene expression data from the exact same tissue slice. And then the final thing to mention is our Cytosist instrument, which is coming next year. This certainly isn't necessary to run the Visium workflow, but it does help. And the premise here is that it will pair tissue that has been sliced onto a standard glass slide with an actual Visium slide within this instrument. So you can go back and access archival tissue slices that you might already have on slides that aren't currently compatible with our product. Or it means that you can kind of go and do your sectioning onto plain glass slides, really look at your sections and decide which ones are of high value to carry forward in the workflow as opposed to sectioning directly on the Visium slide. Just some data from that. We're comparing gene expression clusters detected in FFPE samples that were either sectioned directly onto the Visium slide or with FFPE samples processed by the Cytosist, which is Visium FFP on the top, Cytosist on the bottom. And you can see the same detailed patterns of expression between the different workflows, but the Cytosist simplifies sample handling while preserving data integrity in FFPE analysis. So with that, I want to say thank you very much for today. I know we're butting right up against time, but hopefully we've got a little bit of time for questions, or if not, for Alex to talk a little bit further. But I do want to finish on this slide, which is an initiative we have it between 10x Genomics and Millennium Science that we call Meet with the 10 Experts. So feel free to reach out, email me. We have 3 people here who are direct employees for 10x Genomics, and we're here to answer any questions you've got about single cell sequencing, Visium, from experimental design, right through to budgeting and those kind of things. So always happy to hear from anybody, and feel free to reach out if you want to know anything more today. So with that, I'll say thank you very much, and sorry we don't have much time left, but I'll hand it back to Alex.
Yes, thanks very much, Jeff. And there were a few questions. We do have a couple of minutes, so we might try to address those. So first of all, I'll just read them off for you, Jeff. They're in the chat. First of all, from Paul, what is the sensitivity of detecting cell surface proteins? Could you detect a receptor that is usually about 100 molecules per cell?
That is an excellent question. And I don't, off the top of my head, have the exact conversion efficiency of, if there's so many X molecules on the surface of a cell, how many of them you would find represented in your single cell sequencing experiment. But I will sort of, as a ballpark, say we use reverse transcription to do the barcoding, and reverse transcription itself has an efficiency of, in the ballpark of 30%. So I'd imagine you're probably going to be capturing, as a high level, approximately 30% of those molecules. So maybe 30 your my counts, which can be sufficient for cell surface protein detection. You can use perhaps FACS or something up front to determine, if it's feasible. But certainly, I imagine at 100 molecules on your cell surface protein, it's pretty lowly abundant. You're only going to be capturing a fraction of those, on the order of, at best, 30%.
Thanks, Jeff.
Okay. Secondly, unless you had a follow up on that, Paul.
No, that's fine.
Yeah. All right. And then Caitlin Philby had a couple of questions. Does Cellplex utilize BioLegend hashtags, or are they 10x multiplexing oligo conjugated antibodies? What's the efficiency of Cellplex? I think you may give a part of it. Yeah.
Yep. So we don't use the hashing. That's the BioLegend solution. What we did was come up with our own. It's basically a lipid molecule with a DNA tag on it, so that inserts itself into the membrane of cells. We found that to be more robust than relying on expression of a cell surface protein that you bound to. We've heard anecdotally and seen some evidence ourselves that you can miss certain cell subtypes if you're relying on, again, using an antibody to bind to cell surface proteins to do the hashing, whereas ours inserts itself into cellular membrane. So every cell has a membrane, so it works a bit more robustly. So we also have a full end-to-end solution for our multiplexing, meaning we've got demonstrated protocols on the front end to do the actual sample labeling, and then on the back end, all our cell ranger pipelines sort of do the automated analysis for you in terms of calling cells, identifying your samples, and passing them out into the individual reactions.
Yeah. Caitlin also had a couple of other questions, which were excellent. In the interest of time and letting people get on to their work, I might just suggest, Caitlin, if it's all right with you, we'll follow up with you individually on that, because I did want to just sort of wrap things up and show, just give a quick comment about the fellowship program and how people can participate if they are interested. So let me just see if I can pull up my slide. Hopefully people have my slide that's showing the Hudson Institute Fellowship Program on their screen.
No, I have to share the slide, Alex.
No, bear with me, sorry.
You're right.
Let's have something pop up at the same time. There we go. Have you got all of that?
Yep.
All right. So very briefly, the Hudson Institute Fellowship Program, we touched on at the beginning. It consists of a training program that would enable researchers at Hudson to develop a high level of expertise in single cell or spatial biology technology, and support with development of the specific project that you want to undertake. So there'll be project planning consultations from, as Jeff indicated, our 10 experts. It would also involve a substantial discount on the reagents. So this is one big benefit of the program, that the discounting would be at the highest level offered to any institute in Australia. And there's actually a very easy application process, which is indicated on this flyer here shown on the right, that everyone should have received. If you haven't received a copy of this and would like to inquire more about participating in the fellowship program, you're welcome to contact me personally. My email address is given here. All of the training and the 10x experiments, I'd like to point out, will be conducted in partnership with the Medical Genomics Laboratory run by Trevor Wilson. And so just invite you, Trevor, if you'd like to have a few comments about the status of the installation, and if you have any comments at all about how you'd like Hudson Institute researchers to engage with your core there, I invite you to do that.
Yeah, thanks, Alex. I mean, I guess that most people know that the sequencing capacity we have over here in it, and now we have the 10x Chromium here as well. We've just been going through training with my team here, and so I guess that I'm here on site if you're interested in going further. Obviously, people like Jeff and Kat King of 10x can offer their expertise on things that we're just learning at the moment, but certainly we will work with you to facilitate your experiments, and hopefully a lot of people will be taking up the fellowship program with Millennium and 10x, and we'll work with you every step of the way, I guess.
Sensational. And just as final reminder, should have popped up on your screen, there's a short poll that we would be grateful if you could reply to about the talk and your interest in the fellowship program. If you just take a moment, there are just 2 quick questions on that. That would be highly valued and appreciated. Okay. All right. Karen, can you tell me if I think we've covered everything, and I know we're over time slightly, so we should probably conclude. Are any, if we do that, then it's going to close off response on the poll.
That's fine.
Yeah, okay. All right. Unless there are final remarks from you, Paul, we might conclude there, but I think, thank everybody for attending.
No, that's good. Thanks, Alex. It was very useful, and hopefully give a few people inspiration and momentum to take it on. Thanks, Jeff, for the presentation.
All right. Thanks again for the invitation. Thank you, Jeff, for your presentation, and look forward to engaging with the Hudson Institute in this program going forward.
Thanks, Alex.
All right.